And welcome everybody again to our second and final real tutoring session of new content for the regional sessions that we have coming up, the regional competitions. Just as a quick announcement, the next session is going to be a little bit different. It's going to be sort of similar to the local competition review that we had done, where it's going to be very interactive with questions. We have another set of really great faculty presenters that are putting questions together and going to be presenting those slides on behalf in partnership with the Anato-Bee. So we're really excited for that. In addition to that, you might have noticed on our website that we've shared that there's a couple of different formats of testing beyond just a multiple choice exam that includes our standardized patient and our oral exam as well. And then along with our practice questions, we'll include some image-based and some written ones. So that should cover at least some exposure for all of the different sorts of questions that you can experience at the regional level. So we're really excited for that. There is a recording of something similar done to that last year if you'd like to, again, review what was done last year. But again, we'll have brand new questions for you to work through this year. Another thing is that we are very excited to announce our ABITS recipients, and that will be done tonight at the end of the Zoom call. So hang with us if you guys are really excited to get that announcement. If you're missing tonight for the live feed, just keep in mind that we will be emailing these announcements out as well. And then finally, if you didn't receive any funds and you weren't chosen for the ABITS, but you were really needing that money to attend the regional competition and you're looking at other sources, if you do go to any local groups or communities around you, if it's a community center or at your high school, if any of them are willing to give you funding, we do encourage that you explore those avenues. And we also wanted to say from the Anato-Bee, if you need any documentation on our behalf to prove or sort of support your initiative and to sort of expand on the benefit that you would get from attending, please reach out to us and we're happy to provide you with some of that documentation. So with that, do we have any other announcements or anything that I missed from our Anato-Bee people? All right. So with that, I'm going to introduce our first presenter, and it's an honor to present her because she's one of my great colleagues. This is Whitney Karriger. She's currently an assistant professor of anatomy at the Auburn campus of the Edward Via College of Osteopathic Medicine. Before coming to VCOM Auburn, she taught primarily anatomy at the Alabama College of Osteopathic Medicine at ACOM for 2 years. She holds degrees in anthropology, and yes, that is degrees plural. And anthropology is the study of human behavior, biology, culture, society, and evolution. And that's coming from University of Michigan, Illinois State University, and Tulane University. So no better expert than to talk about anthropology than Dr. Karriger, and we're really glad that she was able to join us today.
Okay, well, thank you so much. I appreciate you inviting me, and I'm excited for this opportunity. So thank you so much for that. I'm sharing my screen, I think. Can everybody see okay? Okay, cool. Is it okay if I go ahead and get started then? Okay, cool. Excellent. Well, today I will be presenting on the cardiopulmonary system. I did this last year as well, so I'm excited to be back doing this again this year, and I'm grateful for the opportunity. So I know this is just a review for you all, but I sort of broke up the learning objectives first for anatomy, and then the histology, and then the embryology at the end. So we'll go through heart and lungs both for each of those things, and we'll start with the anatomy. And so all the learning objectives are here, and then the titles of the slide will sort of tell you which learning objective that information is referring to. Okay, so I know that this wasn't a learning objective, but I just wanted to, just as a quick review, remind you that your heart pumps blood, oxygenated blood out to your body, out to your tissues, out through arteries, out to your tissues to get oxygen. Out to your entire body, and then it flows back through the veins, back to your heart, and then that's pumped out. Deoxygenated blood is pumped out to your lungs so that it can get oxygenated, then it's pumped back to your heart and then out to your body. So that's sort of like the whole system that we're talking about today. And so this is sort of just a review slide for all of those concepts in case you just needed that review. Okay, so the thorax, all the things that we're talking about today, or most of the things that we're talking about today, are really inside the thorax. And so we can divide the thorax really into 3 compartments. 2 of those hold your lungs. So one holds your left lung, one holds your right lung. And so we'll talk about that in a little bit. But first, we're going to talk about the 3rd compartment, which is your mediastinum. And so that's the one in the center here. And that holds your heart and some other structures that we'll talk about on the next slide. So Out here, we have the pleural cavities, but first we are going to talk about the mediastinum right here. And we can divide the mediastinum up further. We can divide it into a superior part up here above this green line, and an inferior part down here below this green line. And so this is showing the same thing up here, but just a lateral view. And this green line in both images is at about the level of the sternal angle. So that level is about how we divide the superior and the inferior mediastinum. And then we can divide the inferior mediastinum further, and you can see that better here in this image. They're sort of color-coded here. So we have an anterior mediastinum here in this orange color, and that really is behind, so posterior to the sternum and the ribs, and then anterior to the pericardium, which is like the sac that holds the heart that I'll talk about more in a minute. And then the middle part of the inferior mediastinum in blue right here. And then the posterior part of that inferior mediastinum. And that really is from the posterior part of this pericardium here back to the thoracic vertebrae. So these are the thoracic vertebrae. So everything in yellow, that's the posterior part of the inferior mediastinum. In terms of the contents, up here in the superior mediastinum, we have the aortic arch. So right here, the aortic arch and its branches. We also have the superior vena cava right here, and tributaries to the superior vena cava. We also have the thymus, which is not being shown in this particular image, but we have the thymus. We also have some nerves. We have the trachea, which is back here. This is the trachea. Also the esophagus. So you can't really see the esophagus in this particular image, and that's because it's posterior to the trachea. So from this view, it's hidden back behind the trachea. And then we also have the thoracic duct, which is part of the lymphatic system. Now, in the inferior mediastinum, again, we have those three different parts. So out here anteriorly in this orange section, we have the thymus and some lymph nodes. In the middle inferior mediastinum here in blue, we have the heart, the pericardium, roots of those great vessels that I just talked about, the pericardiacophrenic artery, and also the phrenic nerve running through the middle part of the inferior mediastinum. And then posteriorly, back here in yellow, we have the descending aorta, the azygous, hemiazygous, and accessory hemiazygous veins, also the thoracic duct and the esophagus, all in this posterior part of the inferior mediastinum. Okay, so I mentioned the pericardium. We can think of if we think of the pericardium as like this balloon here, and this person's fist as the heart, if we were to push the fist or the heart down into this balloon, we come up with this sort of double-walled sac. And so that's how we can think about the pericardium, as this double-walled sac. And this has two main parts. So it has a fibrous part and a serous part. And so here we're looking at a cross section. So this is labeled as myocardium. So that's like the heart muscle. The endocardium is like the lining of the inside of the, in this case, the ventricle of the heart. And so for the pericardium, we really want to be looking out here. So most externally, we have this fibrous pericardium right here. And then we also have a serous pericardium, but there are two layers to that. So there's a fibrous, or I'm sorry, a parietal and a visceral layer. The parietal layer is fused to the inside of the fibrous pericardium here. So that sort of is, when we think of parietal in anatomy, it's usually like the walls on the walls of something. So in this case, it's fused to this like outer wall part of the fibrous pericardium. And then the visceral pericardium right here, that's fused to the heart. So in this case, visceral is referring to the organ. So it's the part that's against the heart. And then in between those two layers of the serous pericardium, we have this a little bit of space here, and it has a little bit of fluid in it, a little bit of serous fluid in it. And this is really important because this helps to cushion the heart. It also reduces friction when the heart beats. So this is sort of like that sac that I was talking about, that's filling up most of the space of that middle part of the inferior mediastinum. Okay, so now we'll just review a little bit about the heart conduction system. So there's sort of two nodes that we want to think about when we're talking about the heart conduction system. We have the SA node and the AV node. And so the SA node, this sort of establishes normal cardiac rhythm. And we can think of this as like the pacemaker of the heart. So over here in this picture, this yellow oval up here, so this is the superior vena cava right here, coming into the right atrium. So this would be right atrium, right ventricle, left ventricle, left atrium. So the SA node is this little yellow spot here, right by the superior vena cava, sort of in the more superior aspect of the right atrium. It will transmit impulses down to this other little spot right here that's being shown in another little yellow oval. That's the AV node. So the AV node is going to get impulses from the SA node, and then it's going to pass those down the AV bundle. So this is the AV bundle here, and it's going to pass those impulses down through the interventricular septum, or that septum, that muscular septum between the right and the left ventricle of the heart. So the impulses will get passed down through that septum and then sort of out to the ventricles of the heart. And this conduction system is really what's making our heart beat. Now, when we think of parasympathetic innervation, generally parasympathetic is referring to things like rest and digest type things, whereas sympathetic things are referring to like fight or flight type things. So like in a stressful situation, your sympathetic nervous system will kind of take over, and that increases your heart rate. So the sympathetic innervation is going to increase your heart rate, whereas the parasympathetic innervation is going to decrease the heart rate. Okay, now we are on to anastomoses. So I know that you've probably already learned about a bunch of different arteries throughout the body. And anastomoses, we can think of them kind of as like interconnections between these different arteries. So you probably learned different arteries, learned their branching patterns to try to create a map of how blood is flowing throughout the body. But we also have these anastomoses, and they're really important because they create redundancy in the blood supply to certain areas of the body. So we have these around joints, for example. So that's what this is trying to show you here, is anastomoses around the knee, anastomoses around the hip. And so this is important because if you think of like flexing these joints, you can be compressing part of those blood vessels. And so you need a way for the blood to still get past this area and out to your tissues. So we have anastomoses around joints. We also have them in the brain, which is being shown here in the circle of Willis. And that's important. It's really important that we get blood to our brain. And so this is just another way for blood to be able to go a different direction if there's a clot somewhere, or if blood flow is blocked for some reason in one area. We also have them in the heart, though. So we need blood flow. Mostly I've been talking about blood flowing through the heart and out to the body, back to the heart and out to the lungs. But of course, we also have a blood supply to like the muscular wall of the heart to keep that tissue alive. And so we have anastomoses in the vessels that are supplying the walls of the heart as well. Okay, so that was all of the learning objectives for the heart part of the anatomy. So now we'll move on to the lung part of the anatomy learning objectives. So I mentioned before that we talked about the mediastinum, which was like the center part of the thorax, and here it's been removed. So it just looks like a big open spot, but remember your heart really is sitting right in there. The other two cavities in the thorax are your pleural cavities. So your left pleural cavity here, and your right pleural cavity would be over here. Kind of like when we thought about the pericardium and it having layers, we also have layers of tissue that line these pleural cavities. So in this case, we want to think about parietal and visceral pleura. So just like with the pericardium, when we had the parietal layer that lined the wall of the pericardium, we have parietal pleura that's lining the walls of those thoracic cavities. And then we also have layers of visceral pleura lining the lungs, so right on the surface of the lungs. And again, like with the heart, we have a little bit of a space in between those two cavities, and there's pleural fluid in there. This reduces friction, and it also helps the lungs to expand and fill with air when you're breathing. And then as a reminder, the right lung has 3 lobes separated by 2 fissures. So that's being shown here. On the right lung, we have a superior lobe, a middle lobe, and an inferior lobe, and that's being separated by 2 fissures. And then over here on the left lung, usually the left lung is a little bit smaller. And that's because the mediastinum is usually offset to the left a little bit. And so the left lung will be a little bit smaller. And we have two lobes, a superior lobe and an inferior lobe here, being separated by just one fissure. In terms of nerve supply for the lungs, we have the phrenic nerve that's really important for supplying the diaphragm. So we need that nerve supply to the diaphragm to be able to breathe. So that's being supplied by the phrenic nerve. We also have some sensory fibers supplying the trachea. So this is the trachea here, the trachea and the bronchi. And those are coming from the vagus nerve and thoracic ganglia. And then we also have, just like with the heart, when we talked about parasympathetic and sympathetic innervation, we have this for the lungs as well. So the parasympathetic in this innervation in this case constricts the bronchi, whereas the sympathetic innervation is going to dilate the bronchi. So just to talk a little bit more about this tracheobronchial tree, because I've been mentioning the trachea and the bronchi. This is trying to show like the branching pattern of the trachea and the bronchi throughout the lungs. So this is being represented by the trachea right here. So this is showing the trachea. And then the first thing that will happen with that is it's going to split into two primary bronchi. So you're going to have a left and a right primary bronchi, one going to each lung. Then the primary bronchi are going to branch into secondary bronchi. There are different numbers of these. So on the right lung, we're going to have 3 secondary bronchi, and that's because we have a secondary bronchus going to each of those lobes we talked about. So the right lung had 3 lobes, whereas the left lung had 2 lobes. So we have 3 secondary bronchi then on the right to go to each of those 3 lobes, and then 2 secondary bronchi on the left to go to each of those 2 lobes. Then after that, we have tertiary bronchi, and then bronchioles, so conducting terminal and respiratory bronchioles, and alveolar ducts and sacs. We can sort of divide this into a conducting portion of the respiratory system and a respiratory portion. So the trachea through the terminal bronchioles, that is all considered the conducting portion of the tracheobronchial tree. And that's because the main purpose of this is really just to move air. So to move air from, as we breathe it in through our mouth and our nose, to move air to our lungs. Then once we get to the bronchioles and the alveoli, then we are in the respiratory portion of the tracheobronchial tree. And that is because this is where gas exchange is happening. So that's what we call this lower part, the respiratory portion. Okay, so now we are on to the histology part of the learning objective. So just like before, we'll do the cardiovascular learning objectives first, and then the respiratory learning objectives next. Okay, so for the cardiovascular part of the objectives, really it's about recognizing veins and arteries in histology slides, and then also different types of blood cells. So we'll go through the veins and the arteries first, and then the types of blood cells next. So when we think about veins and arteries, we want to think of them as like tubes with 3 different layers. And so the 3 layers are called tunica intima, which is the most internal layer, tunica media, which is the middle layer, and then tunica adventitia, or in this image over here, it's called tunica externa. So that's the most external layer. And so this is a cross section here, a drawing of a cross section of an artery. So it's not showing a whole tube, but if this was a whole tube, blood would be in this part here. And we have an artery here and a vein here. And they both have those 3 layers making up the wall of those vessels. And arteries and veins are going to be classified histologically anyway in terms of size. So we have large arteries, large are sometimes called elastic arteries, medium or muscular arteries, and then small arteries and arterioles. And then in the veins, we have the same thing. So we have large arteries, large veins, medium veins, small veins and venules, and then the capillaries over here, that sort of is the connection between the veins and the arteries, because we have the arteries bringing oxygenated blood out to the body and veins returning it. So how the blood is getting from those arteries to the veins then is in this capillary. So that sort of is like the connection between the arteries and the veins. And just in general, in terms of identifying arteries versus veins in these slides, the arteries are generally going to have a thicker muscular wall and a small lumen. And because of this, then the arteries are often going to remain rounded. So it stays pretty rounded. Whereas when we look at this vein up here, they'll often look a lot more flattened. So the wall is still relatively thick in this case, but it has a much bigger lumen or opening in that vessel. And so the veins tend to be more collapsed in these slides. And then I have some more images on the next slide to show you. But in terms of distinguishing large or elastic arteries from the medium or the muscular arteries, the medium and or muscular arteries tend to have a much more prominent internal elastic lamina. And I have a picture on the next slide to show you. But that's what this blue squiggly line is showing here. So that's the internal elastic lamina. It's named internal elastic membrane here. But this is usually much more prominent in these medium or muscular arteries than in the large ones. So I liked these images because it has like an actual histological slide, but then also a drawing with labels next to it so you can understand what's going on. So this is an elastic artery here, a larger elastic artery. It's showing the aorta, and you can see the 3 layers. So we have that tunica intima here, the most internal layer, and then a pretty big tunica media right here, and then a tunica adventitia out here. And so this was that large or elastic artery. Here's an example of a medium or one of those muscular arteries, and you can see this sort of dark squiggly line here. That's that internal elastic membrane that I said is much more prominent usually in these medium arteries than in the larger arteries. This is an example of capillaries just in adipose tissue. So that sort of is, again, the connection between the veins and the arteries. And then over here, we have examples of it's labeled as a cross-section of a vein, but there's actually both in this case. So this is an example of a vein, and you can see how it's more flattened. So it's not keeping that rounded shape as much as the artery right next to it. So this is an artery right here and a vein right here. And then these are the smaller venules and arterioles. And again, you can still see that the vein has a big lumen and thinner walls. And then the artery has a smaller lumen and thicker walls right here. Okay, so in terms of the formed elements of blood, we can think of red blood cells, which you might see abbreviated as RBCs, or you might see them called erythrocytes, platelets, and then white blood cells, or leukocytes. So for this particular review, we're just going to focus on leukocytes because that's what the learning objective was. And so we can divide leukocytes into granulocytes and agranulocytes. And just in general, I guess the red blood cells, their main function is transport of oxygen and carbon dioxide. All of these leukocytes are the white blood cells. Those play a role in the defense mechanisms of your body. So we have granulocytes and agranulocytes, and then we can divide those even further into these different kinds. And I have a picture on the next slide of all of these different kinds to show you. But the granulocytes are the neutrophils. Those tend to be the first white blood cells to arrive at the site of infection. Also the eosinophils. These release histamines and inflammatory reactions. And then the basophils, and these are involved in inflammation and allergic reactions. And then for the agranulocytes, we have 2 different types to think about. So we have the monocytes. These migrate to sites of infection in large numbers. They can also ingest bacteria and cellular debris. And then we also have lymphocytes here, and those appear at infection sites at about the same time as the monocytes, but these are able to recognize specific antigens. So like if you've been infected with a particular infection before, these are what's able to recognize that, and it has sort of an immunological memory. Okay, so these are examples of all of those different types of white blood cells. So this is an example of a granulocyte, and we can distinguish it by looking for this bilobed nucleus. So there's the dark thing in the center here, that's the nucleus, and it tends to have two lobes to it. So a bilobed nucleus with a smooth outline, and it has eosinophilic granules here in the cytoplasm, so that it'll stain pink. And then I just realized that there's some questions, but I go through this and then I can try to go back and answer those questions at the end. So the basophilic granulocyte here also has a bilobed nucleus, but in this case, they're going to be basophilic granules in the cytoplasm. So that's how we can tell these two apart. And then for the leukocytes, there tend to be 2 to 6 segments of the nucleus, but they're also joined together by these strands of chromatin, which you can kind of see one right here and right here. And then for the agranulocytes, we have monocytes. This is an example of a monocyte, and you have a kidney to horseshoe-shaped nucleus here with an irregularly staining cytoplasm, and then a lymphocyte here, which tends to have a rounded nucleus with a rim of cytoplasm around the side. So all of these are the different kinds of white blood cells. These other ones that you're seeing here, those are red blood cells. Oh, my slides aren't changing. There we go. Okay, and then for the histology for the respiratory system, the one way we can tell the larger passages in the respiratory system apart from the smaller ones is that these larger ones, the trachea and the bronchi, that has cartilage in it. So you have cartilage, either C-shaped rings or complete cartilage rings, depending on whether you're looking at the trachea where they're C-shaped or bronchi where there's complete cartilage rings to sort of hold those passageways open. Because remember that the goal of this part of the respiratory system is just to sort of transmit the air, just to move the air. So we can look for cartilage in these slides because we'll have cartilage in these areas and not in these smaller aspects of the respiratory system. But we can also look at changes in the epithelium. So in the trachea, we'll have pseudostratified columnar epithelium, also in the bronchi, some pseudostratified columnar epithelium. But then once we get to the bronchioles, we tend to have low columnar or cuboidal epithelium. And then out in the alveolar ducts and alveoli, we have simple squamous epithelium. So these are some examples of that here. So this is a trachea right here and a bronchi right here. We can see here we can see some cartilage. And so remember, that's in the trachea and the bronchi, we have those cartilage rings to hold the passageways open. So we can see that here. We can also look at the epithelium and see that pseudostratified columnar epithelium. This image here is showing a transition from a terminal bronchiole out to a respiratory bronchiole. But we won't see any cartilage in this cross section here. And then on the next slide, there are some alveoli, and you can see that again we have those simple squamous epithelium that line the alveoli and those ducts. Okay. So now we are on to the embryology part. So again, we'll do the heart first, and then the lungs. Okay, so mostly I've been talking about circulation in terms of adults, where both our heart and our lungs are functioning to get oxygen into our body, pump it out to our body, and then return that deoxygenated blood back to our heart so that it can send it out to the lungs to get more oxygen. But of course, in fetal circulation, the fetus isn't breathing, so it has a different pathway of how the circulation is going to work. So in fetuses, the placenta is really what's going to function as the place for exchange of nutrients and wastes, including oxygen. So we have sort of these three structures that are going to help with this system in fetal circulation that will make it different from circulation in adults. So the first is the foramen ovale here. this is an opening in the interatrial septum. So in the wall that's between the right and the left atrium, there's an opening in fetuses, and it's called the foramen ovale. And this allows blood to flow from the right to the left atrium. And so this sort of helps blood to bypass the lungs, because besides providing blood to the lung tissue of the developing fetus, it doesn't really need to go out to the lungs to get oxygenated because the fetus isn't breathing with its own lungs. So this can help to divert that flow so it's not going out to the lungs. When you're born, the structure is going to close up, and then in adults, the structure, it's like a little depression in the interatrial septum, and we call it the fossa ovalis. Another structure that's important for this sort of bypassing the lungs is this ductus arteriosus. And so this is going to connect the pulmonary trunk and the aorta. So it would be running right in here. We'd have a little connection here between the pulmonary trunk and the aorta. And again, this is going to help blood bypass the lungs, besides the blood that's just going out there to support that lung tissue as it grows and develops. Again, at birth, this closes up. If everything functions normally, this closes up, and it becomes the ligamentum arteriosum. And then the last thing is the ductus venosus. I'm sorry, we just did ductus arteriosus. So this branches from the umbilical vein, and it's what allows oxygenated blood in the placenta to go directly to the heart of the fetus. So that's the ductus venosus. So this is really how oxygenated blood is getting to the fetus. And then this closes slowly during infancy, and once it closes, it's known as the ligamentum venosum. Okay, and then for lung embryology, so the lung is going to ultimately form from the foregut. So we're going to have a lung bud. So that's what's being shown up here. We're going to have a lung bud that's forming from a bulge in the foregut. And this is important to keep in mind, that it's forming from the foregut, because at this point, this is when what will become the trachea will separate from the esophagus. And there can be errors in development that I have on the next slide to show you. But the lung bud is sort of going to bulge out from the foregut and begin that separation from the esophagus. A laryngeotracheal bud will form from an extension of the lung bud, and then the portion closer to the pharynx, that's what's going to become the trachea, and then the more distal portion, that becomes the bronchial buds. And then eventually, the left and the right primary bronchi form. So we're just basically going out this tree in terms of formation of these structures. And over here is like a timeline of when all of these things are happening. But the left and the right primary bronchi are going to form, and then the secondary bronchi, tertiary bronchi, and bronchioles are going to form. Terminal bronchioles will then divide into respiratory bronchioles and alveolar ducts. And then terminal sacs will form in close contact with capillaries, and then eventually mature alveoli develop, and this continues to happen even after birth. Okay. So I think this is the last slide then. Some of those errors in development are these tracheoesophageal defects. So in general, you want the trachea to be fully separated from the esophagus, but sometimes this partitioning doesn't really happen correctly. So this is just 4 different examples of how this can go wrong, but really there are a lot of different examples. In these pictures, the yellow is the trachea here, and the blue is the esophagus. But so what can happen is the trachea or the esophagus can sort of just end. It can have an incomplete connection to the rest of the esophagus, which means there's not really a connection down to the stomach. But we can also have these tracheoesophageal fistulas, which is like a connection between structures where there shouldn't be. In this case, the esophagus has a connection to the trachea. And in either case, this is going to cause problems both with breathing and with the individual getting nutrition. Another issue we can have in terms of fetal development is problems with surfactant. So surfactant's really important for being able to breathe, because it keeps the alveolar sacs from collapsing when you breathe out, basically. And so really, if you're born prior to 28 weeks, Oftentimes those babies will not be able to breathe on their own because they haven't yet developed enough of this surfactant. But that is a sort of another error in development that can occur. I think that was the end.
Awesome job. And again, just really impressed by how much you were able to cover in your 30 minutes. So we appreciate that. Does anyone have any questions for Dr. Karriger about?
I saw a couple. Should I answer the chat ones now?
Yeah, I think we've got one in the chat. I know Dr. Peterson added on to that, but
Oh, she did. Okay.
yeah,
Okay.
we've got, but just for people that are attending on the recording and not able to see the chat. So the question was, why is it that the vein appears more collapsed than the artery? Does that have anything to do with the type of blood they're carrying, oxygenated versus deoxygenated? And then Dr. Karriger, if you want to expand on what Dr. Peterson had said.
I guess I didn't read the entire comment from Dr. Peterson, but it has to do more with the amount of muscle that's in the wall. So arteries tend to have more muscle in the walls of those vessels, and the lumen is smaller, and this just tends to keep it sort of held open, whereas veins they're much thinner walled, and there's not as big of a muscular layer in those veins, and that in these slices causes it to collapse more.
Exactly. Yeah, that high pressure system for arteries, we need a little bit stronger of a wall there. All right, we got another question here is, are macrophages considered granulocytes or agranulocytes?
I'm actually not sure. It's been a while since I don't know. Does anybody else know the answer to that
I'll jump
question?
in, and I know that Dr. Mapes too is an expert on this, but when we think about macrophages, the basic idea is that they are derived from monocytes. So when a monocyte leaves the blood vessel and moves out into the peripheral tissue, we call it by a different name, which is a macrophage. So macrophages are like kissing cousins of monocytes. And of course, monocytes ending in that suffix ending site, just want to point this out, if it ends in that suffix ending site, monocyte and lymphocyte, those are considered agranulocytes. And then if you notice, there's a pattern too with the granulocytes. All of the granulocytes have the "-phil" suffix ending. So maybe that's a nice handy dandy way to remember who belongs in what category.
That's a neat trick. I like that. All right, I got another hand raised. Probably time for one more question before our next presenter. Riva, if you'd like to unmute yourself or put your comment in the chat, either one.
Yes, thank you, Dr. Buskira. So Dr. Karriger mentioned that surfactant helps reduce surface tension. Is that right?
Oops, sorry. It'll increase surface tension. It allows for increased surface tension and basically makes it so that the sacs don't fully collapse when you exhale.
Okay, that makes sense. So is this surfactant like created at birth, or is it something that increases over time? And is there a specific location for it?
I believe I was reading about this a little bit more earlier, and I believe I read that usually between 24 and 28 weeks is when surfactant is really starting to be created. So if you're born prior to 28 weeks, it's often pretty difficult for babies to breathe on their own at that point. After that, there's more surfactant, so there's a higher likelihood of those babies being able to breathe on their own.
Okay, thank you.
Did that answer your question?
Yes, it does.
Okay.
Thank you. Good questions, everybody. You guys are really thinking. All right. Thank you, Dr. Karriger, for your time and your presentation. So I'll go ahead and introduce our next speaker and get Dr. Elizabeth Spudich the floor. So she's joining us again this week, and I'm really happy to introduce her for this session for anyone that may have missed last week's session. Dr. Spudich is the Anatomy Thread Director at the Sidney Kimmel Medical College. That's at Thomas Jefferson University. She earned her Bachelor's of Science in Biology from Chestnut Hill College, and her PhD is in Experimental Teratology and Developmental Biology from Thomas Jefferson University. Dr. Spudich's favorite part of anatomy is whatever she's teaching at that moment. So for today, it's going to be all about the gastrointestinal system, and that's one of my favorites too. When not teaching, Dr. Spudich spends her time with her husband, Sean, and her beagles, Curly Fries and Moody Blue. Still love those dog names, so cute. She likes to knit and crochet, which, as Dr. Karriger just taught us, is a really good way to activate your parasympathetic nervous system and decrease that heart rate. And Dr. Spudich also likes to watch crime shows on TV, which is a good way to activate your sympathetic nervous system and increase your heart rate. So without further ado, I will turn the floor over and welcome another amazing presentation for the next 30 minutes.
Thank you so much. Can I share my screen? Yes, I can. Let me share that. Can everyone see my screen and my giant fluorescent green pointer? Okay. So this is the gastrointestinal system. I was here last week, and I did a whole bunch of things last week, and this is a little bit more focused today. These are the review objectives that we were given. So I'm going to go through them, not necessarily in this order, but you will know where I'm focused because I'll tell you. So we're going to start at the beginning of the GI tract with the structures involved in swallowing. So if we think about swallowing, of course, we chew and masticate and move food around our oral cavity, and then we have to move it into the gastrointestinal tract. And to do that, we have to pass it through a region that is situated behind the oral cavity, which is called the pharynx. So there's a nasopharynx, there's an oropharynx, and there's a laryngopharynx, which is aligned with our voice box. The pharynx is really a common pathway for both air and food or drink. It is guarded partly by two structures. One is the uvula, which hangs down from the soft palate. During swallowing, this is going to be pulled upwards to block off the nasal cavity so that we don't shoot milk out of our nose, although some people can on command. And then as the food is moved down into the esophagus, the epiglottis is a laryngeal cartilage that's going to close and protect our airway, okay? So that we don't choke on our food. The pharyngeal apparatus itself is really made up of a number of muscles. Now, I don't want you guys to be memorizing these very long Latin-based names, but I do want you to understand that there's a series of up and down longitudinal muscles, and then there's a series of circular constrictor muscles. And during the process of swallowing, if we focus down here on the text at the bottom, okay, when we swallow, the tongue is going to move the contents in the oral cavity to the back of that oral cavity. So that's our bolus of food or our liquids. The long muscles are going to pull the pharynx up over the contents of the oral cavity, and then the constrictors will sequentially constrict to move the contents down into the esophagus. And that is how we are swallowing. Okay, so I want everyone to take a deep breath and then swallow some spit. And then think about what these muscles are doing as you are swallowing. Maybe you're eating, I don't know, if you're eating food. I hope you know, whatever you're swallowing, think about what those muscles are doing. Okay, so those are the muscles and structures involved in swallowing. Okay, now once we have moved everything into our GI tract, we need to be thinking about control. So there is a very complex neural control of the gastrointestinal system. The gut tube as an evolutionary organ held our first brain, okay? And that was a series of neurons that now in humans make up what we call the enteric nervous system. This is the brain of the gut. You probably heard about these structures in earlier sessions. We have a group of nerves called the submucosal plexus, and a plexus is just like a braiding of structures. So we have a braid of nerves in the submucosa. We have a braid of nerves in the in between the two muscle layers, and these make up our enteric nervous system. The submucosal plexus is really positioned near the lumen of the gut and is really controlling neural control of secretions into the lumen. The myenteric is controlling the muscles, how they contract to move things along the gut tube in peristalsis, okay? So the enteric nervous system is our baseline control of peristalsis and secretions. And like we just learned with the heart and lungs, we can come in and override that baseline control with our autonomic nervous system. Our vagus nerve is parasympathetic. It's running rest and digest. And this is actually going to increase the activity of this enteric nervous system in response to substances that are in the gut tube. And so that is going to kind of activate our digestion. And then we have our sympathetic, which is our fight or flight. This information is coming from what we call splanchnic or visceral nerves. These are associated with the sympathetic chain and the sympathetic ganglia, and these are going to decrease the activity of the enteric nervous system. When we're running from the saber-toothed tiger, we do not want to be digesting any food, okay? We want to stop that down, focus all of our attention on saving our lives. And don't forget, there's always a sensory component. So if we look at this schematic here on the side, we have the epithelium in our gut tube sensing something is in the gut tube, sending that down to our sensory neurons. Some of that information is going to go up to the brain so that we can control with either the splanchnic sympathetic nerves or the vagus parasympathetic nerves. And some of that sensory information is going to go directly to our enteric system, the submucosal plexus and the myenteric plexus, to either start secretions or start peristalsis at the same time. So that is our neural control of the gut tube. The last thing with respect to the anatomy is I want to talk about the peritoneum and the peritoneal cavity. I was so glad that we just got done that lecture with all the pleura and all the pericardium, because the peritoneum is basically the same sort of structure. When we think about the peritoneal membrane, it is a serous epithelial membrane. It's going to line the abdominal cavity, okay? Just like the pericardium lined the pericardial cavity and the pleura lined the pleural cavity. So the peritoneum lines and covers the peritoneal cavity. It has two components that are continuous with one another. One is the parietal component that is around the walls, covers the walls, and then there's a visceral component that invests and wraps around the organ. In between, there is a potential space, okay? It is probably a little bit larger of a space than what we would see in the pleural space or the pericardial space, because there's a lot of stuff a lot of organs in the abdominal cavity, and they have to kind of somehow fit into the abdominal cavity. But one thing to remember, there are no organs in the peritoneal cavity. The organs are covered by the peritoneum, but do not exist within the cavity. You should never have anything in the peritoneal cavity. Now, with the visceral peritoneum, because there's so much stuff that this visceral peritoneum is covering, some of the organs may be completely covered with peritoneum, and some may only be partially covered with peritoneum. Structures that are completely wrapped in this serous membrane are termed intraperitoneal. And they are things like our stomach is completely intraperitoneal, the liver, the spleen, jejunum, ileum, the transverse colon, and the sigmoid colon are all considered to be intraperitoneal. They are completely wrapped in the visceral peritoneum. The structures that have peritoneum only on one side of them, okay, these are considered to be retroperitoneal. And they include things like the pancreas, the duodenum, and the ascending and descending colons. So here we can see a transverse section. We have our peritoneal cavity, and we have our visceral peritoneum around our small intestine, and our descending colon and ascending colon are only partially invested, only like the ventral surface, okay? And here in this sagittal view, we can also see that the pancreas is not fully invested with the peritoneum, nor is this little component of the duodenum. But almost everything else is completely wrapped in the visceral peritoneum. Any questions so far? Here's my chat box. No questions in my chat box. Michael, you've raised your hand. Do you have a question?
I do. I was curious as far as like, I know the kidneys are retroperitoneal,
Yes.
but does that mean that they're also covered by that visceral peritoneum on one side, or are they something special?
No, they do. They do have a face that is aligned with the visceral, with the peritoneum. They are behind the peritoneum, just like the pancreas is behind the peritoneum. But the kidneys also have a couple of extra wrappings around them. They have a renal fascia, and then some renal fats, and then some more renal fats. And you'll hear a little bit about the special packaging that the kidneys get in, I believe, the next talk. Okay, so they are behind the traditional peritoneum, but they have a couple extra things, a couple extra wrappings going on. Does that make sense?
Would they still be considered would they still be considered a retroperitoneal
Yes.
organ? Okay.
Absolutely.
Okay.
Absolutely.
Thank you.
Absolutely. Okay. So now we've gone through some anatomy. Let's move on to our embryology here. So I took this image from your earlier review from I guess about a month or so ago. Remember, the GI system is from the long gut tube. We have our esophagus, it moves into our stomach, our duodenum, our small intestines, first jejunum, then ileum, then out to our cecum, up through our ascending colon, transverse colon, descending colon, sigmoid colon, rectum. Okay, so we have a lot there. And that large gut tube has 3 divisions during embryology. So we have a foregut, a midgut, and a hindgut. The foregut is our esophagus down to our pancreas, where our pancreatic duct empties into, and everything associated with that. Our midgut is just past that pancreatic duct, so it's the distal duodenum, the rest of the small intestines, and about 2/3 of the way across the transverse colon, and the hindgut is the rest. So we have the distal transverse colon, descending sigmoid, and then the rectum. Now, one of the cool things about GI embryology is that each of these divisions of the abdominal gut tube has its own artery, okay? So first we have the artery of the foregut is the celiac trunk. So it's a large but short stubby vessel that comes off the anterior surface of the aorta. And from that vessel, we have all the vessels that sprout from the celiac trunk are derivatives of the foregut. So we have our left gastric artery supplying our stomach and our distal esophagus. We have our splenic artery, which is supplying our pancreas and some of our stomach as well. We have our common hepatic, which is going to give rise to our liver, supply our liver, still more of our stomach, portions of our pancreas, and our proximal duodenum, okay? So we have all of that blood supply from the celiac trunk or the artery of the foregut. And yes, the gallbladder is a product of the foregut, and I kind of always kind of put that in with the liver. So whenever I'm talking about the liver, I'm also talking about the gallbladder too. I tend to forget the gallbladder. I don't have one anymore. It got taken out, so it's kind of not on my radar all the time. The superior mesenteric artery is the artery of the midgut. So this is a long vessel, again, coming off the anterior side of the aorta, and its branches are going to supply all of the adult derivatives of the midgut that are found in the abdomen. So some of the first branches are the pancreaticoduodenal branches, and as their name implies, will supply the pancreas and the duodenum. We have the middle colic, which is going to supply the transverse colon. Off the left side, we have lots of smaller vessels that are jejunal and ileal vessels, supplying the small intestines. And then off the right side, we have the right colic that will supply the cecum, the appendix, and the ascending colon. The inferior mesenteric artery comes off again. It's another long vessel off the anterior side of the aorta, about 2 inches above the bifurcation of the aorta, where the aorta ends and becomes the common iliacs. This will send branches to supply all of the hindgut derivatives. So we have a left colic branch that supplies the transverse colon and the descending colon. We have a number of sigmoidal branches supplying the sigmoid colon. And then the last branch is the superior rectal, supplying the rectal area. Now, another cool thing we learned about anastomoses in the last session, the last presentation, and so there is a natural anastomosis between the superior mesenteric artery, specifically the middle colic branches, and the inferior mesenteric artery, specifically the left colic branches, that allow for collateral circulation should any of the branches of these 2 major vessels be blocked. Okay, we don't want to block the blood to our intestine, because that would be a really dire situation. Another part of embryology that I know that you guys had talked about has to do with innervation. So when we think about innervation of the gut, we are really looking at, we mentioned before, the enteric nervous system and the autonomics, both parasympathetic and sympathetic. These, as you remember from earlier sessions, the autonomic nervous system uses a kind of midway ganglion. At some point, we have a nerve from the central nervous system synapsing on a ganglion, and then from the ganglion to the target organ and tissue. Now, when we think about that autonomic ganglion, okay, be it sympathetic or parasympathetic, one of the things you have to realize is that all of those ganglionic neurons are derived from the neural crest cell. So when the neural tube was closing, the cells at the very top of that neural tube peel away as neural crest cells. And you've heard all through these sessions how the neural crest cells contribute to all sorts of different structures in the human body. The neural crest cells are going to collect as the sympathetic ganglia, okay, of the sympathetic chain. Some of them are going to collect as pre-aortic ganglia. We'll see them in a minute. And these pre-aortic ganglia are going to control the sympathetic innervation to the entire gut tube. And those neural crest cells are also going to migrate into the gut tube and give rise to the 2 subdivisions of the enteric nervous system that we just talked about, the submucosal plexus and the myenteric plexus. So everything's really all kind of coming together here. We talked about the blood supply and how each of the major vessels off the front of the aorta was an embryonic vessel to part of the embryonic gut tube. Well, the same goes with the ganglia. So when I talk about the pre-aortic ganglia that is controlling the gut tube, these ganglionic cells are going to come and situate around the celiac trunk, the superior mesenteric artery, and the inferior mesenteric artery, and they will give rise to similarly named parasympathetic ganglia. So we have the celiac ganglia up here around the celiac trunk, the superior mesenteric ganglia around the superior mesenteric artery, and the inferior mesenteric ganglia around the inferior mesenteric artery, okay? So here we line up our blood supply with our sympathetic autonomic innervation. The rest of the parasympathetic innervation is really that first is from the vagus nerve, and then the ganglia again are the enteric cells in the organ of the gut tube. Okay, so let's do some histology now of the gut tube. We have our, again, from our previous, we have our gut tube has 4 layers. We have a mucosal layer, a submucosal layer, a muscularis externa, and then a serosa, which is an adventitia that is wrapped in that visceral peritoneum. But as we move along the gut tube, each region is going to tweak that layer to facilitate the function. So let's take a look at some of that tweaking. In the esophagus, one of the major tweaks is the mucosa, is a stratified squamous epithelium where we have multiple cell layers that protect that tissue, protect that organ against abrasion. Remember, we're swallowing things, hot coffee, spicy foods, and we don't want to damage the tissue too much. So we have a stratified squamous epithelium. We also see some tweaking in the muscularis layer. The upper third of the esophagus has striated muscle, and this helps us to facilitate swallowing, but it also helps to facilitate vomiting. While the lower third is just the smooth muscle and completely under the control of the autonomic nervous system. We have an outer longitudinal muscle and an inner circular muscle, and then that myenteric plexus is going to be right in the middle. In the small intestines, the job here is to absorb. So we maximize our surface area available for absorption. The first thing we do is we send folds of submucosa into the lumen. These are called circular folds, or if you rather the Latin, the plicae circularis. Each of these folds has a number of mucosa that has been folded in and out. These are intestinal villi and intestinal crypts. So they come out and they go in and they go out and they go in and they go out and they go in. Within this villi, we have some epithelial cells that are have lots of microvilli for absorption. Some epithelial cells are goblet cells for mucus secretion that lubricate and protect. And some deep in the crypts are for bacterial immunity, okay? And these are called Paneth cells. So if we take a look at what these look like, here in this upper left panel is a nice histological section. We see our plicae circularis. We see our villi coming off each of those plicae. We move to this panel here in the upper right. This is a zoomed in on that villi. We can see all of our enterocytes that have our microvilli brush border. And here's a nice goblet cell releasing its mucus into the lumen. And in this bottom panel, we can see at the base of an intestinal crypt, these cells, these are Paneth cells, and they're filled with what look like red gumballs. And each of these gumballs is holding a bunch of antimicrobial substances to keep the bacteria out of your gut, the bad bacteria. In the colon, we have no circular folds, no villi, no Paneth cells. Our muscular layer, we have our 2 muscular layers, but our mucosal layer is really just set up for water absorption and mucus secretion for lubrication. So here we can see lots of colonic crypts here. We can see mucus cells here, goblet cells secreting mucus, and all the rest are there to absorb water from the stool. The appendix is, we all know, is a small diverticulum that kind of a worm-like appendage that hangs off the cecum. It's kind of a little bit, half of it looks like colon, half of it looks like small intestine. The mucosa is simple columnar cells, a lot of mucus secreting cells there as well. We have our 2 muscle layers on the outside, but the hallmark of the appendix is the submucosa is riddled with these large lymphoid nodules. And it is believed now that these lymphoid nodules help set up the adaptive immunity for the entire gut tube. In the stomach, we have a number of adaptations as well. The muscularis external layer actually has 3 layers of smooth muscle. There's a deep third layer that is an oblique layer. And the 3 layers make sure that the stomach can move in all 3 planes, and we can adequately mix our food. The myenteric plexus is still located between the first and second layer, just like it is in all the other regions of the gut tube. The mucosa of the stomach is folded into gastric pits, and then they expand out into kind of tubular gastric glands. The cells lining the lumen of the stomach and the neck of this pit are all mucus-secreting cells. They're all protective. Remember, we have a lot of acid in the stomach. We want to protect those tissues. So that mucus is there to neutralize the acid and the gastric enzymes. That acid and gastric enzymes are really coming from the gastric glands, which have 4 different cell types, certainly mucus for protection. The parietal cells are the acid secreting cells in the gastric gland. The chief cells down here at the bottom of the gastric gland are the enzyme secreting cells. So these are going to secrete pepsinogen. And then sprinkled along the gastric gland are enteroendocrine cells, and these provide a local hormonal control. They secrete things like gastrin, ghrelin, and motilin to help prime the rest of the GI tract when the stomach is full. Okay, our last set of histology, looking at the accessory organs of digestion. So we're going to start with the liver. Liver cells are called hepatocytes, and they are ranged in cords. So these are like long columns of cells. Each of these cords are fit into a 6-sided plate, okay? And then the plates of cords are stacked around and aligned with the blood vessels in the liver. And each of these, and a cluster of these stacked plates is called a hepatic lobule. That lobule has a central vein, okay? And the outside, the points of that hexagon, okay, each point has a little bit of the portal triad, which we now know, hopefully from earlier sessions, to be a portion of the hepatic artery, a portion of the portal vein, and a portion of the bile duct, okay? So here is a schematic where we're looking at just kind of one corner of our hepatic lobule. We can see that the blood is moving from the portal vein and the hepatic arteries, moving towards the central vein. The blood is filling these hepatic sinusoids. It's percolating through all of the hepatocytes. And as the blood is percolating through the hepatocytes, they are detoxifying. They're taking the nutrients out and metabolizing them, the sugars. They are metabolizing the hemoglobin that's being dumped from the spleen into the blood, and certainly getting oxygen from the hepatic artery. As they're doing all this, they're also making bile, and bile is moving away from the central vein into the bile duct. So the blood is moving in one direction, and the products of the hepatocytes, the bile, are moving towards the opposite direction. The gallbladder, I didn't completely forget the gallbladder. The gallbladder is really adapted for storage, concentration, and controlled release of the bile that is made by the liver. So we have a mucosa that is a simple columnar epithelium, and it's really set up to concentrate and absorb fluids from the bile. The muscularis externa is very, the muscles are arranged haphazardly, again, so that the bladder can contract in all directions. so that it can effectively push out the bile when needed. And then we have the pancreas. The pancreas is two glands in one. We have an endocrine gland function where we are secreting hormones directly into the bloodstream. This is from the islet cells, and the two major hormones that are being secreted there are insulin and glucagon. And then we have an exocrine function that is secreting digestive enzymes, and these are going directly into the duodenum, and these are being secreted by these glandular acini out here on the periphery. And we make about 1.5 to 3 liters of pancreatic fluid per day. So that's a lot of fluid being pumped by that pancreas. And here we have a histological section. We can see our islet cells right here. These are the endocrine portion pumping out insulin and glucagon. We can see our little rosettes of secretory acini here around the islet cells. And here's a little part of the ductal system that is going to be taking the secretions of this acini and moving it into the gut in the duodenum. Okay. And that is it. And if anybody wants to see, these are my two puppies. But I will take questions. What are the questions already in the chat? Okay. The non-oblique, so there's always a longitudinal and there's always a circular in the gut tube. So there's a longitudinal layer, then there's a circular layer, and in the stomach, we add an oblique layer, okay? So that we have one going up and down, one going around, and then a third at different angles, okay? So that we can get not just a contraction, not just a shortening, but like a twisting as well when we move those muscles. Do the hormones released by the enteroendocrine cells in the stomach help activate? So those hormones are really, they're activating the enteric nervous system. That's probably their primary target, to prime like the very next region of the gut tube, but they will circulate as well and have effects on the central nervous system. That signaling is very, very complex. I don't want you to get too bogged down on it, but I want you to know that those cells, when the stomach is full, they are saying, hey, we need to prep for what's coming down the pipe. So they're sending out very local hormonal signals to the next section of the GI tube. Let me just see here.
Looks like we've got a question from William. I just wanted to say thank you for that presentation. That was very thorough and again, very condensed for what you
Very
tried.
condensed.
Very good. Very good
talk all
representation.
day.
Yeah, I know, right?
Who wants to unmute themselves first?
I think William had his hand up first. We'll let him go
Yeah,
first.
so did we ever say what the purpose of bile was?
Oh, so bile is to emulsify fats and lipids and cholesterol.
Does it
So
is that what emulsify means?
yeah, so so remember the lumen is largely a water-based, so the gut tube inside is largely a water-based system. And in order for the fats to be effectively metabolized, they have to be because they'll separate out from the water, just like a salad dressing. Okay, the bile helps to kind of put a charge onto the lipids and allows them to remain in solution so that they can be taken up by the cells of the gut tube and moved into the circulation. Does that make sense?
Yes.
Who else?
That would be me. That was a good question.
Who is me?
Michael.
Hello, Michael.
So the you mentioned the liver picked up hemoglobin from the blood, does it, and you said like this, the spleen deposited it. So does that mean the spleen destroys or like dismantles the worn out red blood cells and then releases
Yes.
the wastes, and then the liver picks up the wastes and metabolizes them?
Absolutely. You're
Okay,
right.
cool.
You're bang on. So the red blood cells, the ones that are tired and have been used a lot, they are broken down by the spleen. They're dismantled. They're chopped up by macrophages, and the reusable parts, like the iron and the hemoglobin, are sent back to the liver, and the liver is going to process them. And that's a whole big, it's going to be a whole big set of biochemical reactions at some point that you guys are going to love to learn. But you're absolutely right. The spleen breaks down, sends the iron and stuff back to the liver to be processed and reused. And then, and part of that, what can't be reused is moved into the bile.
Well, actually, how does it know which red blood cells are like worn out and like ready to be recycled, and which are like still strong?
So as the cells are moving through your body, they are picking up other proteins on their surface. They are sometimes getting a little jagged. And the spleen, the macrophages in the spleen will see the other proteins that are stuck onto the surface of the red blood cells and pull them out. So that's a very simplified version of how we get rid of red blood cells.
Would that larger amount of proteins also slow them down when they're trying to go through the spleen?
No, it doesn't tend to slow them down. It may make them a little bit, like I said, more likely to be recognized by a macrophage as kind of a little tired.
Okay, thank you.
Yeah. I think we could go on all day. My email's at the front of this. Email me and we'll talk all day about red blood cell clearance.
All right, looks like we got another question from Riva. Do you want to go ahead and unmute yourself?
Yes, thank you. So you might have already mentioned this, but I was wondering what part of the red blood cell can be salvaged?
So there's actually so the red blood cells are going to be taken up by the macrophages, okay? And most of what is there is recyclable in some fashion, but it's chopped up and then it's going to be kind of used and kind of like broken down, almost like a garbage disposal. Okay. And then a lot of that stuff will go back kind of into the fluid of the blood, and it will be like the individual amino acids will be reused, the individual components of hemoglobin will be reused. So it'll circulate back into the bone marrow where new red blood cells are being made. So it's a very, very complex process. But just like every cell in your body that is used up and removed through apoptosis or just an old cell is going to be kind of taken in, chopped up, and then all of its component parts, all of its proteins, amino acids, nucleic acids will eventually be recycled. Our body doesn't waste a whole lot. It would be inefficient.
Excellent. Thank you so much. I think we're going to go ahead and move on for our next speaker to make sure we get through those. If you guys still have questions, feel free to keep putting them in the chat, and we can try to still address those for you. But I'll turn things over to Dr. Peterson to introduce our next speaker. And thank you again, Dr. Spudich, for a lovely presentation. Very informative.
Great.
All right, well, feel free to please put things in the chat if anything comes up. And moving forward, then I'll introduce our next speaker, our final speaker for the evening. This is going to be Dr. Kathryn Veazey. She's from Colorado Springs, Colorado. She graduated from Colorado State University with her bachelor's and master's degree in biomedical sciences, and then she completed her PhD in clinical anatomy at the University of Mississippi Medical Center. She's a part of the anatomical sciences teaching team at Drexel University College of Medicine, working alongside Dr. Dana Peterson. And when she's not teaching, Dr. Veazey enjoys reading, painting, and playing tabletop role-playing games. And I just love that. And I'm so excited that your creativity can, that you have outside of the classroom. It was also able to come into the classroom, as we saw from your presentation that you gave for us last year. So we're really excited that Dr. Veazey was able to rejoin us again this year. So without further ado, Dr. Veazey.
Happy to be here. Hello, everybody. Very excited to talk to you. I'm talking about one of my favorite things today, which is the genitourinary system. Let me pull up my little timer so I don't yammer away. All right, let's get started. So here are, oops, it's not advancing. Let me try again. There we go. Here are your objectives for today. So I listed them in the order that they were listed with the anatomy. You can note that all of them are numbered. So when you go back and review this, each slide has the number objective at the top of it that it will be relevant to. So we will be first talking mostly about the renal system. We'll actually be first talking about how we even build a body and its cavities. And then we will finish off with some reproductive system, including spermatogenesis, folliculogenesis, menstruation, the mammary gland, lots of things. Quick caveat, whenever talking about reproductive, genital, or pelvic anatomy, I do think it's fair to discuss just briefly the difference between sex and gender. So for my purposes in this talk, be aware that when I use the term male, that means someone who was born with the anatomy that is suited to creating sperm, versus when I say female, that means someone born with the anatomy that is suited to creating eggs or ova. So if there are male associated structures, those are associated with spermatogenesis, so something like a testis or a penis, versus female associated structures are those associated with oogenesis and folliculogenesis, so things like an ovary, a vulva, the uterus, etc. Be aware that none of those things are gender, which of course is much more complicated, more of a social construct, very socially determined. And so I will really just be focusing on sex-based characteristics and not gender. So, first things first, everyone has a body. We split them into cavities. So our body cavities, we can see here, we first have a dorsal body cavity, and this is actually going to be split further into the cranial cavity, which will house our brain and our brainstem, and then continuous with our vertebral cavity. And the vertebral cavity is what will actually house our spinal cord. The ventral cavities, and remember, ventral just means towards the stomach. So for our purposes, we can also say the anterior cavities are going to be subdivided into the thoracic cavity, which is going to be inferiorly bounded by our diaphragm. And in this purpose, it's going to be our respiratory diaphragm, because we've actually got just below that an abdominopelvic cavity, which is inferiorly bounded by its own diaphragm, the pelvic diaphragm. So two diaphragms that we're really talking about here. Within the ventral body cavity, superiorly, we've got the thoracic cavity. The thoracic cavity is then further subdivided into the mediastinum, which we already heard about today in the very first talk, so I'm not going to spend too much time on this. Remember, the mediastinum contains the heart as well as the lining of the heart, which is the pericardium. Just to either side of that, we have the pleural cavities, which will then contain our lungs. Below, we can see that abdominal cavity, which is continuous with the pelvic cavity. These are arbitrarily divided by certain landmarks that we'll talk about in just a moment, but be aware that the entire space is continuous. A lot of those organs are moving between both the abdomen and the pelvis. The whole thing is lined with peritoneum, with some structures being either behind it, retroperitoneal, or actually below it, subperitoneal. So the way that we develop these cavities is, remember, all the way back in early week 3, we have just started the process of gastrulation. We are forming some tissues, and late into week 3, sort of right at the beginning of week 4, we are going to start the process of body folding. And by the time we started this, note, we already have started forming a very primitive heart. So we can see it marked here as the heart prominence, which is right near our developing head and our developing throat. So this we can actually see the pharyngeal apparatus with this heart starting very close to it. So as we begin our folding, we first do craniocaudal folding. So we go head to tail folding. And this takes our little embryo from a flat disc into sort of a peanut shape. And that's going to allow us to create some of these cavities, but also migrate them down. So we need this heart, which was all the way up, very cranial, to move away from the throat and down into the very beginning of the pericardial cavity, so that it will be in the thorax where we need it as an adult. As this is happening, we will also start forming the cavities lateral to it, which will be the pleural cavities, and continuing inferiorly to the peritoneal cavity. The next thing that's going to happen, we can see here in C, we're going to have lateral body folding. So we're taking our little peanut disc and we're actually wrapping it in on itself. So when we do this, we're going to form the primitive body cavity, which is called the intraembryonic coelom. Coelom is just a primitive cavity. The process of lateral body folding is going to let us create what we call our tube within a tube model. So we've got this little primitive gut tube. Remember that we've got this umbilical vesicle, also referred to as the yolk sac, which was sort of, at this point, not really herniated, but think of it like it's kind of herniated out away from the wall. So we're actually going to sort of come in and pinch this closed later on in the process of body formation. As we do this, if we move a little bit further towards development, we can see we've got somites developing dorsally. Remember, those are going to help us sort of segment the portions of our vertebral canal. More ventrally, we can see we've got this pericardial cavity that is separated now from our developing gut, labeled here as just the midgut and the hindgut. We can see the foregut more cranially, and those are separated by this little ring of pink, which is the septum transversum. We can see it here in the cross-sectional view as well, wrapping a variety of tissues, including centrally our aorta. We can see once again that developing gut tube here, and now our primitive peritoneal cavity. This pink tissue, that septum transversum, is pretty much the main component of that respiratory diaphragm. However, we can see that the diaphragm is going to have several additional contributions to it. So looking now at this image, just a heads up, this image does fully flip your orientation. So be aware that in image C and F, the dorsal part or the back of the embryo is where my cursor is towards the top of the image. However, now we can see that the dorsal view is actually at the bottom in B, C, and D. We can see this because we can see our little vertebra right here with the ribs growing out from it. So ventrally towards the stomach, here is that septum transversum, which is really going to be forming the vast majority of that respiratory diaphragm. Just posterior and lateral to it, these are what are called pleuroperitoneal folds. If you break those roots down, they actually tell you a lot about what they are. So the pleuroperitoneal fold is going to be a little bit of an outpouching, a growth that is separating the pleural cavity, which is the lungs, from the peritoneal cavity, which is again sort of that initial abdominopelvic cavity. So these are going to grow towards the septum transversum and close to fuse. While all that is happening, our little gut tube is starting to differentiate. So we've actually got a primitive esophagus, and that esophagus is associated with its own mesentery. That esophageal mesentery will also contribute to the very central portions of our diaphragm, especially helping form what is called the central tendon, which is the very shiny part right at the middle where the heart sort of sits. Last but not least, developing from the body wall itself, we're actually going to have just a slight growth inwards that we can see right here, sort of in this purple color, lavender almost. And that's a little bit of muscular ingrowth from this developing body wall. And those are going to form the portions of the diaphragm that actually come down and reach and connect, sort of anchoring on laterally to that rib cage. So once we've formed a body, we've got to finish it at its most inferior end, its most caudal end. And this is where we actually have that pelvic diaphragm. For our purposes, I'm just going to call it the pelvic floor. Know that there are technically differences between the diaphragm and the floor. However, we're not going to focus on those for this talk right now. But what we can see in this view, we're actually looking from a bird's eye view down into the pelvic bowl, and we've removed all of the organs out of it. So we can see just the musculature and some of our orifices that are going to be opening up externally towards the perineum or the groin region. So the purpose of the pelvic floor is a lot. It's doing almost everything for us, actually. It's going to help keep everything above it where it's supposed to be. And just think about how many organs that is. That's going to be the entirety of your abdominal viscera as well as all of your pelvic organs as well. It's going to help resist increases in abdominal pressure. So when you cough, when you sneeze, when you strain in any way, it's once again going to help keep things where they are and prevent what's called prolapse, which is when things can actually go through these orifices and exit out from their appropriate cavity. And then I would argue for most people in day-to-day life, the one we can think about the most, they're going to help us maintain continence. So these are what allow us to choose when we use the bathroom. There are many components. First, we have what is called the levator ani. These elevate the ani, which is the anal region. They're actually doing a lot for us. They're not just really focused on the anus. They're actually going to be helping us with the more anterior orifices as well, which include things like the urethra and the vagina if it is present. It has 3 components, which you are not responsible for, but just be aware, we can actually see there's sort of a U-shaped sling around these orifices. And then as we move laterally, we can see a slightly bigger U, and then sort of a triangle a little more posteriorly. So all of that together is the levator ani. They have extensive connections starting anteriorly from the pubic bone, moving around towards the ilium, and anchoring posteriorly towards the coccyx. Just posterior to that, we have a new little triangle, and this muscle is simply called coccygeus. Some people will call this ischio-coccygeus. That's totally fine. That is because it connects the ischium to the coccyx. All of these are innervated by some nerves, specifically some ventral rami, off of your sacral plexus. So we're really looking at the very, very, very bottom of the spinal cord, levels S3 through S5, roughly. So now for a good pretty much third of our talk, we're going to be talking about the renal system. And I like to talk about it development first. I think that's very beneficial. Remember, we need to form a kidney. A kidney is the little bean-shaped organ. We've got one on each side. They're sort of posterior in the abdominal cavity, and they're tucked right underneath that respiratory diaphragm. The left one is going to be a little bit higher because our liver is taking up so much space on the right-hand side. These are going to be developing from our intermediate mesoderm. And it's worth noting this is really going to be starting pretty early in the 4th week of development. So we've formed our body cavities, we've got our heart going where it's supposed to be, and we are actually going to transition through 3 stages, which we can see here. So the first thing we do, starting pretty cranially actually, we are forming what is called the pronephros. Whenever you see neph, N-E-P-H, you should always be thinking kidney. And that's because the functional unit of the kidney is the nephron, which we'll be talking about extensively in the subsequent slides. This pronephros is really just a scaffold that's going to sort of create the primitive systems that the kidney will then build upon. So the pronephros forms and then regresses very quickly. It is going to be taken over by the mesonephros. The mesonephros we can see is this nice pink sort of ridge here, and it's got a whole bunch of little tubes forming in it. Again, that scaffolding having been provided by that pronephros. The mesonephros is the first functional kidney of the developing embryo. Excuse me. It is going to regress partially. However, it's also going to have contributions to the reproductive systems, especially in male-associated reproductive development. In everybody, regardless of sexual development, we're going to form this little sort of nub right here. This is a diverticulum. Remember, a diverticulum is just a fancy word for a little bud. And this is called the ureteric bud. Ureteric bud because it's going to help us form our ureter. And our ureter, remember, is going to be sort of the main excretory tube that will connect our kidney down to our urinary bladder. So once we formed our ureteric bud, we can now form our definitive kidney, which is the 3rd stage of the kidney. And this is going to be the metanephros. The metanephros will form what is called the metanephric blastema, which is actually going to be induced to form by signals that are actually coming from this ureteric bud. It's a very cool process. It's very complicated. I'm not going to talk about it extensively right now, but just understand we sort of form this little bud. It talks to the tissues around it, and then those tissues form this blastema, and they will actually grow towards each other and branch extensively to form this primitive functional kidney. The ureteric bud itself will form the ureter, but also the structures that will drain directly into the ureter. So think of your collecting structures. So these are going to be everything from your collecting ducts, through your calyces, through the ureter itself. The metanephric blastema will create all the other components of the nephron, except for the blood supply, which we'll talk about in just a second. And last but not least, we connect all of these tissues down from that ureter into our developing bladder. However, when we are first, again, very early in development, thinking late week 4, transitioning into weeks 5 and 6, we've got what is called a cloaca. A cloaca is an undifferentiated sort of all-purpose opening. It is used for excretion as well as insertion in several species. However, we are going to septate this cloaca. We're going to partition it with something called the urorectal septum. Once again, breaking your words down, urorectal septum, it's going to break us into a urinary portion and a rectal portion, which we can see happening here. At that point, we will now have a definitive urinary bladder, which we connect to that ureter, and then posteriorly a separate tube for solid waste excretion, being the rectum. So, how do we excrete waste? Well, we're fetal. Well, first things first, that mesonephros, which is the second stage of our kidney development, does work. It's actually going to start forming waste. It's going to pretty much be done with that role by about the 8th week of development. At that point, the metanephros is going to take over, and it will continue its function all the way until it's fully a kidney, and it will continue that function until, hopefully, the duration of your adult life. So we will be forming urine while we are in utero. So we're going to actually excrete that urine into the amniotic fluid. And we do this in one of 2 ways. We are either going to just put the urine into our bladder and excrete it out through the urethra. However, we can't do that if we don't have a urethra yet. And there are stages in fetal development where we actually haven't formed that opening yet. So instead, what we're going to do is go up through this little tube here, which is called the urachus. And to get into semantics just really quick, this tube is actually called the allantois, and then that's when it's going to be open and hollow. It's actually going to close and form a little cord, and that cord is called the urachus. However, it can remain open, and what this means is we're going to connect the bladder to the belly button. And so there is an amount of waste that will actually be excreted through the umbilical region, through the umbilical cord, really, into that amniotic fluid. That should close up and allow normal excretion to be occurring just through the urethra. We don't really need to worry about solid waste as a fetus because we're not taking in any solids. And so it's actually sort of a big deal once the fetus is born. It will actually pass its first stool, and this is called meconium. And that's going to actually give you a lot of information about sort of the health of that fetus. Once that waste is produced, swallow it to recycle it. And so the balance of waste and liquid production should be maintained by the rate of actually swallowing that tissue and all of that fluid. So moving on to the nephron itself, we can see a full depiction here of not just the nephron, but actually the full uriniferous tubule. The uriniferous tubule has multiple components. They include a blood component. Remember that there's going to be a very key player here, which is the glomerulus. The glomerulus is sort of a ball of yarn. That's what glomerulus refers to. So it looks like a little wad of capillaries, and that's where we're going to be able to pull things out from the blood into our filtration system. So we enter the nephron proper, which is going to be composed of the Bowman's capsule. The Bowman's capsule plus the glomerulus is what we refer to as a renal corpuscle. Once we move out of that corpuscle, we will enter our proximal tubule system. So proximal is just close to the corpuscle. We will first have a convoluted portion, and then we will have a straight portion. You may see the straight proximal tubule referred to as the thick descending limb of Henle. That's totally fine. That's because it leads us into our thin limb of Henle, also referred to here as just the loop of the nephron. As we come back up, we have a thick ascending limb, also called the distal straight tubule. That will transition us into our distal convoluted tubule, and then we're done with the nephron. The nephron will actually connect to the collecting system, which, remember, had a different embryological origin. So once we connect here at this collecting duct, we have actually now connected our metanephric blastema structures to our ureteric bud structures. From the collecting ducts, they will all go out towards our ureter, so we can then bring that urine down into our bladder. Looking at these tissues histologically, we can see first, this is all going to be from the renal cortex. We can see the actual images and then compared drawings over here. So the renal corpuscle, again, is sort of a ball of string. So it's got a core of glomerular capillaries, and then you can actually see the simple squamous lining outside of it, which is the parietal layer of Bowman's capsule. Much like the tissues that line cavities, so the pleura, the pericardium, the peritoneum, we're going to have a visceral layer, which will line the innermost tissues, and a parietal layer that lines the outer tissues. So the parietal layer here is very distinct. The visceral layer of Bowman's capsule is actually lining all of these capillaries themselves. And they have a very special cell type. It's actually called a podocyte. They've got little tiny feet that are going to help them with the filtration of fluid out from the capillary and into this urinary space here. So we can see those once again here in this drawing. As we look just to the side of this, we can see our proximal tubule. Proximal tubules are very acidophilic, also eosinophilic, either one's fine. Remember, that just means they stain very intensely and they stain sort of a very bright pink color. These have a brush border, which means that they look sort of fuzzy. They also sometimes have what's called a stellate lumen. We can actually see that depicted over here as well as over here. So it's sort of star-shaped. It's not very circular. It's not very oval. It's got really sort of a characteristic appearance. And those are going to be true of whether it is a convoluted tubule or a straight tubule. As we look now to sort of this orange-yellow box, this is going to be a distal tubule. Distal tubules are a little bit more pale staining, although in this particular image, they're very similar. But we can see over here, it's slightly different. So these are going to be a simple cuboid layer. They do not have a brush border, so the lumen should be a little bit more open, a little bit more patent, and it shouldn't be any kind of particularly irregular shape. Last but not least, we will have our collecting ducts. A collecting duct has sort of one main characteristic that is going to help you identify them, which is fairly distinct, what we call intercellular boundaries. This means that you can actually see the lines between each adjacent cell. So we can see here, here's a line, here's a line. We can see them not all the way, but most of the way around. We can actually see some that were not highlighted in the box here. So here's some lines. Here's some lines, so on and so forth. If we move away from the renal cortex, which is that outermost layer, we can go down into the renal medulla, which is the core of our kidney. The tissues here are still going to be the collecting ducts. So collecting ducts go all the way through the kidney, connecting us from the very pretty much top of the cortex all the way down to our final excretory system as we start to form those calyces and out into our ureter. However, this is also where we'll see now our loops of Henle, as well as their associated blood supply, which is called the vasa recta. So prior to this, our tubes are mostly going to be simple cuboidal. So this is true of our proximal tubes, our distal tubes, and our collecting ducts. However, both the loop of Henle and the vasa recta are now transitioning to simple squamous, which we also saw in the Bowman's capsule. So remember, we've got that little parietal layer of simple squamous tissues. So collecting ducts look exactly the way they did previously. We can see those distinct intercellular boundaries, nice sort of circular to oval lumen. And if we compare that to something like this, this is now simple squamous. I don't see a lot of cytoplasm. I see a pretty open lumen. And I can also see the nuclei here should sort of bulge in a little bit to the lumen. So it almost looks scalloped, not quite. But it is going to be a bit more distinct from this little box right next to it. So I can once again see really only one nucleus in this example, but it's pretty flat. It's sort of smushed. It's not going into my lumen at all. And so that's going to be characteristic of a vasa recta. In this particular example, it's kind of nice because the vasa recta should be a little bit smaller. However, depending on where you get your section, the way that it's been cut, that's not always going to be the best option. The best thing you could see is actually some red blood cells in those vasa recta, because you shouldn't have any blood in your tubule system, provided you've got a healthy kidney. So continuing on, talking about how all of these tubes work, here we can see the uriniferous tubule, no glomerulus present. We've just got the sort of excretory structures. So first things first, our Bowman's capsule, its whole job is to move things, which we call solutes, from our capillaries into the ultrafiltrate, which is the fluid that we make before it becomes urine, which is its final form. So the priority of the proximal tubule system is all about reabsorption. This is why it has a brush border. So remember, a brush border is just a series of microvilli, and their whole purpose is to take pretty much maximize surface area to move things into their lumen. So we've got our brush border, so we can do most of the absorption. We can see not only are we absorbing the most things, but we are also just absorbing the most volume-wise as well. So almost all of the reabsorption is going to happen, at least initially, here in this proximal tubule. As we move down into our thin limb here, this loop of Henle, this is where we're going to do this very cool thing where we concentrate the ultrafiltrate. So remember, we've got those vasa recta, those little arteries that are running right alongside of it. We're going to do something called countercurrent exchange, which is when the flow through the nephron is in the opposite direction of the flow through the vasa recta. And in this way, we're actually going to be moving solutes and water in different amounts at different places through different regulatory mechanisms so that we can have an incredibly hyper concentrated ultrafiltrate at the very base of this loop. And then we can fine tune it as we move through our distal limbs. So we can see here, this is where we're really going to be sort of in the adjustment phase. We're going to do a little bit more resorption of some of the things we've already done our initial reabsorption of. Last but not least, as we enter into this collecting duct, that's our final chance to figure out how much fluid do we really need to be getting rid of, how much urea do we need to be getting rid of versus keeping in the body. Because once it's past this point, it is fully urine. It's going to go out into the ureter. It's going to be excreted through the bladder. The way that this whole system is innervated is pretty cool. So I'm actually going to start from the bottom and move up. So we would like to have voluntary control of this system. And the way to have voluntary control of something is you need somatic nerves. Remember, somatic nerves are just to typically like body wall and limbs. So skeletal muscle gets somatic innervation. So we do actually have some skeletal muscle associated with our urinary system. And this is a muscle called the external urethral sphincter. A sphincter, remember, is just sort of a small hole that opens and closes. So this is going to be a little tightening muscle that actually compresses the urethra. And so we have voluntary control of this muscle. It's controlled by a nerve called the pudendal nerve. The pudendal nerve is sort of the main nerve of the genital region. It's going to help us with both urinary retention and continence, as well as fecal continence. And it allows that sphincter to be relaxed on command. So this is actually a reflex we have to learn as we get older. This is the nerve that you train with potty training. We've also then got our involuntary control mechanisms, and these are all about those autonomics. So here we can see a little diagram sort of depicting the sympathetic nervous system. Remember, sympathetics are going to start in your spinal cord between the levels of T1 through L2. For urinary control, we're mostly looking at the L1, L2 levels. These are actually going to come out as a series of splanchnic nerves and synapse in that inferior mesenteric ganglion, which we've already seen in the previous talk. From the inferior mesenteric ganglion, they will actually form a series of nerves, which are called the hypogastric nerves. The hypogastric nerves are going to pretty much suppress or inhibit the contraction of the bladder, which is a smooth muscle, to allow it to fill. So they don't want you, again, if you're thinking of the sympathetic nervous system, it's fight or flight. If you're running from a bear, you don't really want to pee during that process. So you're going to inhibit that. So you're really going to allow relaxation of the bladder to allow it to get bigger. Parasympathetics, however, these are going to be innervated by a very special kind of nerve called pelvic splanchnic nerves. And those cell bodies actually originate in the spinal cord as well, but they're originating between L2, 3, and 4. Depending on what you read, which textbook you're using, those levels will be different, but most people agree S2 through S4 are sort of the main levels of that. So those preganglionic cell bodies are going to come out and travel as what are called pelvic splanchnic nerves, and then synapse into ganglia within the wall of the bladder to stimulate it to empty. So the actual muscle of the bladder itself will squeeze. And this is when you feel the urge to go to the restroom. So we're going to transition from this point into the reproductive systems, first starting fairly close to the bladder. We're actually starting within the testes. So the testes are associated with spermatogenesis. Spermatogenesis is the male-associated type of gametogenesis. Remember, gametogenesis is how a sex cell is going to undergo its own process of division, which we call meiosis. For the timing of this talk, we're not going to go into all of those processes, really just talking about the different components, what they are, and where you can see them. So first things first, spermatogenesis is happening within the testis. If you cut into the testis, you'll see a series of tubes, and these are called seminiferous tubules. Seminiferous, they're helping produce sperm, which will then be mixed with a sort of proteinaceous and sugar-filled fluid, which is semen. So that's where it gets the name seminiferous. It's got a lumen, which is where we're going to put the mature sperm cells, but it's also got what we call the adluminal compartment, which is away from it. And we can actually see that that's going to be bound by a membrane out here. So as we go through the process of spermatogenesis, we're going to start in this adluminal compartment and then move closer towards the lumen. So we can see this nice sort of black and white image, and then we can also see them in light microscopy and in the illustration here as well. So starting off, we have what's called a spermatogonium. The spermatogonium is in the most closest sort of basal part. They are very small, they're very round. There's not a lot going on with a spermatogonium. So we can see some examples here, right? They're pretty big, pretty nondescript, really nothing super exciting happening. Spermatogonia are going to divide and create what are called primary spermatocytes. Primary spermatocytes are also big. They're actually pretty massive compared to most of the other cell types we're going to see. These are going to be the biggest type of developing sperm cell that we will view. And they're fairly heterochromatic. Remember, heterochromatic, they're going to have a really, really, really dark nucleus. So we can see lots of examples here in the light microscope, these really big, really dark cells with just a thin rim of cytoplasm around them. Those are your primary spermatocytes. Those are going to divide into secondary spermatocytes, and secondary spermatocytes only exist for a brief instance. And so they're actually really, really difficult to see in microscopic views. What we can see, though, is the consequence of their maturation, which is to actually form spermatids. And spermatids have two subtypes. We have early spermatids, which are very close to the lumen. They are going to be still pretty heterochromatic, so they're really, really dark, but we're going to start forming our classical sperm shape. So we're going to have sort of a little triangular head and a little tail, but early spermatids don't have a tail yet. That is what actually distinguishes them from a late spermatid. And so early spermatids can be seen very well in this view. Unfortunately, there's no really good examples of late spermatid in the black and white image, but we can see some down here. So if we look at like the light microscope view, I'm going to try, it's really hard not to cover them up with my cursor. I'm sort of circling right now. These are some early spermatids right here. And we can see some late spermatids right nice and pointy, right at the edge of this lumen, getting ready to get released out into the seminiferous tubule itself. All these little strings that we can see getting left behind, those are those tails. Because sperm cells or spermatids are an incredibly motile cell. They've got to move through reproductive systems to fertilize an egg. So if we look at the comparable process, this is the process of folliculogenesis. Folliculogenesis is the development of cells around an oocyte. Remember from gametogenesis, oogenesis and spermatogenesis are following the same basic steps, but they're pausing at different points. And so when we're talking about the actual preparation to ovulate a follicle, there's only one of two kinds of oocyte that we really care about. So we're never going to see the oogonium, and we're also never really going to see a mature, fully fertilized ovum, because it actually finishes meiosis after fertilization happens. So really only two types. You've either got a primary oocyte or you've got a secondary oocyte, and the secondary oocyte only exists right before ovulation happens. So instead of focusing on the oogenesis, we really focus on the folliculogenesis. So first things first, we have a primordial follicle. This is the very beginning stage. We can see our oocyte or our egg, and it's covered by a single layer of, at this point, not very well differentiated cells. These are called granulosa cells, and they're pretty much squamous, and there's nothing else going on. So just really one layer of granulosa cells. We will then continue to develop into a primary follicle where we've still got one, maybe a few more, say 1 to 3 layers of granulosa cells, but we've transitioned now from simple squamous into simple cuboidal. As we move out further into the secondary follicle, we can still see our singular little oocyte, and now we've stacked on a few layers of granulosa cells, and we've now started to form a new cell type around it, which are called theca cells. We may also start to see the development of what is called the antrum, which is a little fluid-filled space that is much better seen here in a tertiary follicle. A tertiary follicle, depending on how big it is, most times it's still going to have a primary oocyte in it, but right before it gets ovulated, it will have a secondary oocyte. Because again, the process of meiosis will resume right before the egg leaves the ovary. We can see now we've got a lot of layers of granulosa cells. They actually start forming some specializations, including this little stalk and this sort of ring or crown around the oocyte, which is called the cumulus euphorus. We can also see a lot of layers of theca surrounding the outside of this. We have this large antrum, which again is just a fluid-filled space. And then last but not least, this tertiary follicle, which is the most mature form, will be finally ovulated. So it will be sort of ejected out from the ovary. The follicle will actually rupture. The fluid that was in that antrum will be released. And some, but not all, of the granulosa cells will follow along that little secondary oocyte as it leaves. What is left behind in the ovary is something called the corpus luteum, which we'll talk about more on the next slide. Excuse me, the slide following. So here we can see all of this in pictures. So these are all images of the ovary specifically taken out. Remember, most organs, we're going to have an outer cortex and an inner medulla. So we can see here in the ovarian cortex, we've got a lot of primordial follicles, which really just look like one egg and pretty much nothing going on around them. They've all got a thin rim of cells, just that single layer of squamous granulosa cells. However, we can see the development of one primary follicle. So it's gotten much bigger, and we can actually see, even at this magnification, a sort of thicker ring of cells just outside of it. As we continue on, we enter into our secondary follicle. We can see the egg still larger. We can now see some granulosa cells around it. And not shown great, we do have some theca cells just starting to form. The tertiary follicle is going to have an egg. We can't see the nucleus because it wasn't caught in this particular section, but be aware it does still have a nucleus. We can see the granulosa with their specializations, as well as a lot of thick layers outside of it. And then we can actually see 2 types of theca. So a very light type right here, this is called theca interna, and sort of almost an adventitial looking layer, which is the theca externa. The follicle cycle is going to actually be linked to our ovarian cycle. So this is all part of what is called the menstrual cycle, which is something going on in a very complicated process relating the uterus to the other tissues like the ovary. And I know my time is short, so I am going to go through this very quickly. So we know we've got our primordial follicles going all the way through our more mature follicle at the tertiary state. As these follicles are developing, that takes roughly 1 week. So this is the follicular phase. Once that dominant follicle is selected, it will be ovulated, and the development of that singular mature follicle takes another 7 days. So ovulation occurs at day 14 of the cycle, and after this, the cells that are left behind are, again, that corpus luteum. So these are going to mostly be those granulosa cells, as well as that little layer, 2 layers of theca cells that surround it. The corpus luteum is actually going to produce a lot of progesterone, which is there to help maintain pregnancy if it should happen. If pregnancy does not occur, the degeneration of the corpus luteum will actually stimulate the initiation of the menstrual cycle, which is when the uterine layer starts to slough off. So here we can see the histological structure of that corpus luteum. So we first have what are called granulosa lutein cells. So they've undergone a reaction that makes them different. It's called luteinization. They are very big, they're very pale, and they produce a lot of hormones, the main ones being estrogen and progesterone. We've also got our theca lutein cells. These are very small, they're very dark, they're more compact. We can really see that in the zoom out here. And these are producing androgens, which are a more masculinizing hormone, as well as progesterone. It's active for a few days, but if fertilization doesn't occur, it will just completely go away. If fertilization does occur, it will maintain the pregnancy until such time as the placenta takes over. The uterine cycle, briefly, is going to be linked to that ovarian phase. Remember that the uterus has two layers. We have a basal layer that stays, and then we have a functional layer, stratum functionalis, which is going to be involved. So we can see we've actually got menstruation, which is the sloughing of the stratum functionalis. We've got proliferation, where we start building that layer back up, and we have the secretory phase, which is characterized by a variety of glands that start to develop. We can see all of these on the next slide. Each of these is taken at the same magnifications, low mag at top, high mag at bottom. We can see this functional layer during the menstrual phase. It's pretty small. We can actually see pieces getting sloughed off into the uterine lumen. During the proliferative phase, we start to build up. We have a series of pretty narrow, straight glands. And then we can see the secretory phase, we have what we call saw-toothed glands. So very big lumen, sort of all over the place. And it's absolutely very, very, very thick. Hi, everyone. Briefly, we're going to review the stages of external genital development. So we can see over here in the table, there are three main structures that we're going to be concerned with. First, starting off with the genital tubercle. The genital tubercle is going to be indicated by hot pink in the image over on the side. We can see here, first, the undifferentiated genitalia, which is primarily dominated by what we call the primordial phallus. The phallus will become the erectile tissues. We can see it becoming differentiated on the right-hand side of the image with the female-associated development, and on the left-hand side with male-associated development. The phallus in male development is going to be the glands of the penis, as well as most of the tissues of the penis. In female development, it was going to form the glands of the clitoris, as well as a series of erectile tissues called the bulb of the vestibule. The bulb of the vestibule is going to be directly deep to or underneath the skin and tissues of the labia minora. The labia minora are coming from our second portion, which is called the urogenital fold, which is sort of this peach color in the image. They're labeled as urethral folds in the picture. Either term is completely fine. We can see it here forming those labia minora. We can see that further developing. Here in male development, we can see it's forming the ventral surface or the underside of the penis. Last but not least, we have the labioscrotal swellings, and those names will once again tell you exactly what they're doing. We're going to help form the labia, this time majora, or the scrotum. So we can see labeled in sort of purple or lavender in the images. We can see laterally in female development, forming the labia majora. We can also see the development of the scrotum just below the phallus or the penis. The development of these tissues is regulated by a series of hormones. So first things first, in female-associated development, you typically have XX chromosomes, which are missing a very important gene, which is called SRY. The absence of SRY is actually going to cause the gonad, which at this point is undifferentiated, to develop into an ovary. When an ovary is created, it will start regulating the release of estrogen, and we will have the absence of masculinizing hormones, which include a hormone called anti-Müllerian hormone, which is going to actually inhibit the production of female-associated structures, as well as testosterone, which would stimulate the development of male structures. And therefore, we will have the development of what are called the paramesonephric ducts. The paramesonephric ducts are going to contribute to most of the internal reproductive structures that we associate with women. After the internal structures have developed, we will continue to form the external structures that we just saw on the previous slide, especially, again, the clitoris, the vestibule of the vagina, which has the bulb of the vestibule, and the labia, both minora and majora. Note that the absence of these masculinizing hormones will cause the mesonephric ducts, which are formed by that mesonephris, which remember was our second stage of kidney development. And if with the absence of the mesonephric ducts, we do not produce a hormone called DHT. DHT stands for dihydrotestosterone and is also a masculinizing hormone. So if we look at the male-associated pathway, we are going to follow very similar, but now, instead of having estrogen and the absence of masculinizing hormones, we will only have masculinizing hormones. So most male fetuses are going to be XY for the chromosomes, which means that the Y chromosome will have our SRY gene. The SRY gene will trigger the gonad to differentiate into a testis, and that testis will trigger the production of both testosterone and anti-Müllerian hormone. Anti-Müllerian hormone will cause the paramesonephric duct to regress. The other name for the paramesonephric duct is Müllerian duct, which is where this hormone gets its name. The presence of the testosterone will cause those mesonephric ducts to differentiate into male-associated structures. The external structures will be triggered to develop by the presence of DHT. DHT is the major hormone that regulates secondary male sex characteristics. So these are things like facial hair, but also, again, external structures like the scrotum and the enlargement of the penis. It is worth noting that DHT is a main player in sexual ambiguity. So here we can actually see the Quigley scale of genital ambiguity, which is rated on a scale of 1 through 7, although only 6 are typically shown. And we can see that in the presence or absence of certain hormones, the genitalia can go through a variety of stages of ambiguous forms, because regardless of what kind of gonad is present, it is actually the hormones, especially this DHT, that regulate these external genital characteristics. Mammary glands, these are a modified sweat gland. So it's a combination of a merocrine cell, which remember is going to pack stuff into little vesicles before it releases it, as well as apocrine cells, which form their product and then put it into the very apical portion of their cell, and then it sort of blebs off. It's more fatty, it's sort of foamy. The whole purpose of a mammary gland is to produce milk. Anybody with mammary glands is capable of producing milk if the right hormones are involved. It does not only occur during pregnancy or after pregnancy. There are several components. We have the ducts, which contain the milk. These are called lactiferous ducts. They've got a simple cuboidal lining. They are further supported by a variety of connective tissue and fat. We can see the secretory portion of the gland. This is the alveoli. And each of these alveoli typically is associated with a specialized contractile cell called a myoepithelial cell. Their whole job is to squeeze the alveolus to put the milk out into the duct system. When your mammary gland is inactive, it is almost entirely connective tissue and fat. We can see very few alveoli sort of scattered around, and they're very inactive. They're entirely alveolar. And talk about the external genitalia development as the last sort of thing later. I apologize for going over, but happy to answer questions.
Thank you for, again, I'm just amazed at how great of a job all of our presenters do at shoving a lot of content into a short period of time. So Dr. Veazey, thank you for making these really complex materials really understandable, and really appreciate that. We do have a couple of announcements. As promised, we said that we had the ABITS for last, so I'll let Dr. Haggerty do that, and then we'll do our closing remarks.
Hello, everyone. Can you hear me all right? Awesome. Okay, so I just wanted to let you all know that I will first say thank you to all of our ABITS applicants for all your hard work in essay submissions. I will say that this year's ABITS award winners, this was made possible due to the Department of Undergraduate Medical Education at Thomas Jefferson University. They are sponsoring the ABITS winners this year. Each winner, there are 3, are going to be given up to, or being reimbursed up to $1,000 for travel to the competition. So without further ado, I would like to name the first 2 because I did see them on the call tonight. So one of our ABITS winners is Emily On. I don't know if I saw a few people hopping off the call. I think she might have popped off, but we'll give a congratulations to Emily On if you want to throw up a little emoji or anything like that. Our next winner, which I see they're still on the call, is Shunjoy Maji. So congratulations. And then I did not see their name tonight, but Crystal Martinez is also one of our winners. I was fortunate enough to, yay, you are so welcome, Shunjoy. You'll be getting an email from me tonight. So if you have any questions, feel free to reach out. Crystal Martinez, like I said, is our 3rd winner. She came to the Eastern region, so I did get to meet her last year, and she'll be here again. So congratulations to all of our ABITS winners. Thank you everyone else who applied. You'll be receiving correspondence from me, and I'll let Dr. Piscuri take it away with the rest of the announcements.
I mean, I can't end it any better than that, but I'll just say congrats again, like Dr. Haggerty said, and just want to remind you all that our final tutoring session is our review session. And remember, again, like we said at the beginning, that we're going to do that a little bit different. We're going to be covering those SPs and our oral examination and kind of the format for that. And then like we had done with the local competition, we will have the opportunity for participating in taking these questions and polling you guys so that you can get a sense for where you're at. So we do very highly encourage you to attend that session. And just as a reminder, that is 1 week from today. That is February 23rd at our usually scheduled time, depending on what your time zone is. So that's all I got. Any other things from any other anatomy folks? All right, we're good. Everyone have a great evening and we'll see you next week.